So
Okay.
I have just started recording.
Okay, thank you. You are once again welcome to the UDIS webinar series. And today, as usual, we are going to make a presentation, and our speaker of today is Professor Umar Imam, who will talk to us about the center he heads, that is CAMRET, Center for Advanced Medical Research, Center for Advanced Medical Research, that is now growing to be a world center. And I don't want to say much about the center because the topic of today centers around it. We are going to hear about the center from the director. But before I give chance to the director to present, I want to say a word or two about the director himself, being the leader for today's webinar. Professor Mustapha Umar Imam, as you have all know, holds a medical degree from Bahrain University, Kano, and a PhD in molecular biotechnology from the University of Putra, Malaysia. After his PhD, Professor Mustapha also started working as a postdoctoral fellow at UPM from 2013 up to 2015. He later joined Zhengzhou University in China as an associate professor, of nutritional biochemistry, up to 2019 when he joined the services of Usmanu Danfodiyo University as a reader. Professor Mustapha Imam was elevated to the rank of professor in 2023, and he is the director of the center and also the grant management officer of the university. Professor Mustapha has won several research grants, both locally and internationally, and he has been ranked consecutively for 4 years as top 2% scientist by Elsevier. I think without much waste of time, I will invite Professor Mustapha to start the presentation. Prof, can you please share the screen?
Okay. So can you all see my screen?
Yes, sir. We can see it.
So is it moving?
It is moving, sir.
Okay. So thank you very much, Dr. Mani, for that a very wonderful and concise introduction on myself. So just like Dr. Mani stated, I am the director of the Center for Advanced Medical Research and Training, CAMRET, which is one of the numerous research centers at Usmanu Danfodiyo University Sokoto. And the purpose of this presentation today is to give you insights as to what we do at my center. And while I give you insights into what we do, I'll give you ideas on, I'll talk to you about what we've been doing in the past couple of years and what our potentials are and where we see ourselves getting to in the next couple of years into the future. So I'll just quickly go through my presentation since we're already starting behind schedule. So the outline for this talk is going to be, as you can see on the screen. I'll first talk about the university in general, very briefly introduce the faculties and talk about the faculties and the centers in the university. Then I'll narrow down to CAMRET, give you an overview of CAMRET, the laboratories we have, talk to you about some of the equipment that we have at the center, what research projects we've worked on in the past and what we're currently working on, what has been our major source of funding at the center, and what our strengths and challenges are, and what our future potentials are at CAMRET. So Usmanu Danfodiyo University Sokoto, which my center is a part of, has 3 major campuses. The main campus where the administrative center, which is the administrative center of the university, and a lot of faculties and departments are situated in the main center. in the main campus of the university. Then there's a city campus, which currently majorly houses the veterinary teaching hospital and a few other centers. And then we have the teaching hospital campus where medical and allied health science courses, faculties and courses are. And that is where CAMRET is located in the teaching hospital campus. So in total, the university has about 17 faculties as at now, and then it has a total of 11 research centers to complement the teaching services, the teaching and academic services at the at the various faculties. Hello, can you hear me?
Yes, sir, you are loud and clear.
Okay, because my network appears to have. Okay, fine, it's okay. So the university also has about 11 research centers to complement the academic and teaching activities at the 17 faculties around the university. So CAMRET is one of those research centers and arguably the most vibrant. And I'll share with you in the next couple of slides what we've been doing, and then you can judge for yourself if there's any other center in the university that is as vibrant as CAMRET and or has been as productive as CAMRET over the last couple of years that CAMRET has been very active. By the way, CAMRET itself was first it was commissioned in 2017, and then I took over the headship of CAMRET in 2019. And then when I took over CAMRET, already there were a set of mandates that the center was already built on. So the center was actually mandated to do research and development programs related to molecular biology, genomics, cancer biology, neglected tropical diseases, genomics of complex diseases, nutrition-related chronic diseases, immunobiology and vaccinology, cardiovascular physiology, and diabetes. So unfortunately, what we met on ground in 2019 was a very dismal situation at CAMRET, whereby although the center had a set of mandates, but there wasn't any proper direction in terms of research or any services that the center the center had. And then we also met a situation whereby there wasn't any plans on ground to ensure sustainability of any research or any service that was done at CAMRET. So one of the things that we did was to not only begin to engage solidly in research, but we also ensure that whatever we did, we will keep a blueprint, and we will make sure that there will be some sort of sustainability to what we're doing. So some of the first, for some of the first few steps that we took were to actually ensure that the center had a befitting website. Now, whatever you do, if nobody knows about what you do beyond the four walls of your center, then chances are that nobody would really understand that you're doing very well, and people wouldn't really appreciate that you're doing it. And perhaps somebody out there might even be needing the services that you offer, but they wouldn't really be aware that you have those services. So they wouldn't know how to contact you. So we made sure that we had a befitting website. We had ways that people could contact us. And these, for example, I have noticed over the years that a lot of our centers or a lot of agencies in Nigeria would have a contact number belonging to an individual, whereby if that person leaves, then it's difficult for it's difficult to then get in contact with that particular center or agency. But so one of the things that we've tried to do is to have a dedicated phone number to for CAMRET, so that even if I leave or any other person that is currently in CAMRET leaves, there'll still be a way to reach reach out to CAMRET. And in addition to that, we also made sure that we had social media presence all over the major social media sites like Facebook, LinkedIn, Instagram, and Twitter, which is now X. Now, we've actually significantly seen increase in traffic to our website, and we've also seen increases in the number of inquiries that we've gotten and the number of requests for services through from our social media sites. because prior to this time, I mean, prior to establishing these social media accounts, it was difficult for people to actually reach out to us. But then now that we have these, it's we constantly give updates on what we do, what happens in CAMRET, what are some of the services that we do, and then sometimes we do have vacancies for employment. We have research. We have positions for research assistants, for research officers. We also have positions for studentships, MSc, PhD. We do advertise them on our website, and we get a lot of feedback and a lot of inquiries through our social media sites. Now, when you check on, okay, so I'll talk to you about the laboratories that we have, and this is directly coming from our website. So if you log on to our website and then you go to our laboratories, you'll see that our laboratories are categorized into the general laboratory, general biotech laboratory, where we have what we mostly do in this general biotech lab is this is where we mostly store our samples. We have our refrigerators, we have our freezers, we have a -20, we have various -20 degrees freezers. We have two -80 degrees freezers. We have, interestingly, we also have a -150 degrees freezer, which is arguably one of its kind in the country. I may be wrong, but I have not heard of any other -150 degrees freezer across the country. We also do some wet lab experiments here involving mostly chemicals. For example, we do have some students that extract bioactive compounds from plants. This is where this is the lab that they use for their general experiments. And then we do have a proteomics lab. This is where we do most of our protein experiments. So we have this lab has a fluorescent microscope. We do have a flow cytometer. We have an ELISA plate reader. We have protein gel electrophoresis system. We also have an infectious pathogen inactivation lab. Now, the reason why this is important is because we actually have a lot of projects currently ongoing that have to do with handling infectious pathogens. And for example, when we worked on, we were the first lab. CAMRET was the first lab that was activated to handle COVID-19 testing in the entire Sokoto, in Sokoto, Kebbi, and Zamfara states. And in fact, CAMRET was actually the go-to lab for activation of other centers in these three states. So we leveraged on some of these labs that we had, like the infectious pathogen lab, to ensure success of our COVID-19 testing, and then to ensure that we don't have any cases of infection of our researchers or anybody working in our lab. Because we majorly do most of the work that we do is using nucleic acids, DNA and RNA. We have a dedicated DNA and RNA extraction lab. In this lab, we either do manual extraction of our nucleic acids, or sometimes we do an automated extraction procedure. The automated extraction procedure is not, we don't always utilize this because of the cost implication. It's far more cheaper to conduct a manual extraction procedure than an automated extraction procedure. But then at the end of the day, what is important is to ensure that the quality of the nucleic acids that you get for any experiment is top notch. So in this case, we have a nanospectrophotometer. In fact, we have two nanospectrophotometers that we use to check the quality of our new extracted or purified nucleic acids before we use them for any experiments. We also have a pre-PCR room. Now, this is more or less like a master mix preparation room where we do where we mix our reagents before we resort to doing PCR or any other genomic experiments, which are usually done in our genomics lab. This is by far our most important lab in the center, the genomics lab. We have a couple of PCR machines, the conventional PCR, the real-time PCR machines. We have next generation sequencers, the second generation, the third generation. We have an Illumina iScan microarray machine. And then we also have a gel documentation system in this lab. And like I said, this is by far our most important lab, and most of the experiments that we conduct in the center are mostly done in this lab related to PCR, sequencing, gel electrophoresis, and the like. And the likes. We also have a dedicated Drosophila lab. Now, why this is a very important lab for us is because we know that traditionally, when researchers want to model diseases to test certain compounds that they have, to test, for example, interventions that they want to that they want to experiment. Most times, people resort to, at the preclinical level, people resort to the use of animals like rats, like mice, like guinea pigs, rabbits. But then the reality is it's a bit expensive conducting those type of experiments, and especially considering the situation we're in currently in Nigeria, where funding is not forthcoming, and then most times researchers are forced to take money out of their pockets to sponsor their experiments. Which is something we're actually changing. We're changing the narrative in terms of that in CAMRET, and I'll talk to you in a bit how we're doing that. So, but this Drosophila lab, conducting experiments using Drosophila, which is the fruit fly, is significantly cheaper than it would cost somebody to do experiments on lower animals like rats, mice, guinea pigs, or rabbits. And since we've established this lab, I think this was from 2019. Since we've established this lab, we've actually seen significant uptake in terms of the utilization of this lab from undergraduate students, MSc students, and even PhD students, because it's far more cheaper to actually do experiments on Drosophila. So, and very importantly, in this lab, we have a dedicated incubator that keeps our fruit flies. That keeps temperature and humidity within a certain range, so that experiments using Drosophila are quite reliable and reproducible. And importantly, it also has a mechanism whereby it rotates between day and night, so that it could replicate the natural cycle, the natural light and dark cycles that we have for the Drosophila. So we have also an electrophoresis lab. And we also have a tissue or cell or tissue culture lab, arguably one of the most functional in the country, if not the only functional in the country. We do experiments mostly using continuous or secondary cell lines that we purchase from outside Nigeria. So currently we have a stock of cell lines that we maintain. We have had requests from other labs across the country that do try to do cell culture, but then most times we send them these cell lines and they are unable to maintain these cell lines. But we've been successful in maintaining these cell lines. So we do have a lot of our projects currently using secondary cell lines, especially those that are cancerous, liver cancer, breast cancer, cervical cancer, glioblastoma, which is a form of brain cancer. We have leukemia cell lines and a whole lot of other cancer cell lines that we use to study biology of these cancers. We used to study effects of different drug interventions or interventions using different types of extracts. So in terms of the projects that we have worked on, this is coming from our website. We have, if you go onto our website, you will see a list of projects that we have worked on in the past and those that we are currently working on. So, for example, in the past, we have worked, this was from during the COVID period, when we got a special intervention from TED Fund to work on identifying potential therapeutic agents for COVID-19. And this was majorly, this project was majorly done in CAMRET. So we looked at this project was based on repurposing drugs that were currently in use for other diseases, like cardiovascular disease, diabetes, and antibiotics and the likes. And we did some bioinformatic analysis to see which of the drugs would have better potential from using in silico data against COVID, against the virus that causes COVID-19. And we saw a couple of those that had very good hits, which we did wet lab experiments on in our tissue culture lab too. sort of complement the dry lab data, and we got very interesting results which are currently published. We've also worked on a project, again, TED Fund project, which is on the use of a particular plant extract from Ocna cubensis to see if it would be beneficial in the management of glioblastoma, which is a form of brain cancer. We've worked on a project. This was I was the PI for this project on anticolon cancer of a particular compound coming from a plant extract. We also have worked extensively on how low zinc levels, this is majorly using Drosophila, how low zinc levels influence or program for the development of chronic diseases in offspring. And what we try to do with these type of studies is to do transgenerational experiments to see what happens if a parent generation is exposed to a particular factor, in this case, low zinc levels. We've also done similar for low iron levels. What happens to 3 or 4 generations of offspring? And what we've seen consistently is that in these experiments is you restrict zinc or iron in a parent generation, and you tend to see very beautiful, or you tend to see very beautiful effects in the generations down to the third or the fourth. We also actively work on how rice influences consumption of rice, that is the white rice that is commonly consumed, versus the brown rice, which we tend to call in Nigeria the local rice, how that influences the development of cardiometabolic abnormalities in offspring whose parents have been exposed to the rice, but the offspring don't get to consume the rice. We're also currently working on, we do a lot of experiments on development of recombinant DNA vaccines. So we've done that with Lassa fever, we've done that with COVID-19, and we're also currently doing the same with, we're using the same approach to develop a recombinant DNA vaccine, which will be used as an anti-snake venom. So in this case, we're trying to develop a DNA-based vaccine, which if injected into individuals would produce antibodies that would neutralize certain snake venoms. In this case, we're targeting one of the commonest snakes, that is one of the commonest snakes in Nigeria, Echis ocellatus. Okay, I've talked about the recombinant DNA vaccine that we've worked on for Lassa fever, for COVID-19. We also have, okay, good. Another interesting work we're doing in the center is the one that you see, number 19. We're actually in the process of number 11, sorry. We're actually in the process of developing a qPCR kit, a PCR kit that will be able to screen for different causative pathogens of fever of unknown origin. And the reason why this project is important for us is we've actually had discussions with our colleagues in the clinics, and we've noticed that they do see a lot of patients that have fever, and most times they just tend to manage these cases as either malaria or typhoid. And even if these patients don't respond, most times they just say this is resistant malaria, when in fact other pathogens may be responsible for the fever. And in fact, we're currently working with our collaborators from Germany. They have developed a kit that is used to screen for a particular pathogen that causes relapsing fever, which would give you the same presentation as malaria or typhoid in most cases, as we see it in Nigeria, especially because of the abuse of antibiotics and other antimalarials. So the interesting thing we've observed from this particular study is that we have screened patients that have presented to the teaching hospital and then the specialist hospital in Sokoto. We've screened them, those that have presented with fever, relapsing fever that keeps recurring. We screened some of these patients, and we got a prevalence of up to 20% for Borrelia, relapsing fever caused by Borrelia species. So what this means is that a lot of the cases of fever that are being labeled as resistant malaria or typhoid may in fact be things like relapsing fever caused by Borrelia species, or even other types of fevers caused by other pathogens. But because we do not have the diagnostic capacity, or sometimes we don't even check for these pathogens, patients tend to suffer a lot. And at the end of the day, these patients don't get the best care, and then some of them may even succumb to their illnesses because they don't get the proper treatment. So these are some of the pictures. I tried to put some of the pictures, but of the equipment that we have, but because there's a lot to put, I just put a few of them. This is our minus 150 degrees freezer. We have a water system room. Okay, the reason why having a water system room is important, which I forgot to talk about earlier, is because now we know that one of the major issues in science generally or research is the issue of reproducibility. You do an experiment, or you see an experiment and you want to reproduce it, or you want to build upon it, and then sometimes you're unable to reproduce it because the conditions under which that experiment was conducted in another lab cannot perfectly be recreated in your own lab, or some of the conditions were not truly reported as they were conducted. So one of the ways that you could actually try to standardize your experiments to overcome some of those issues with reproducibility is to have to use water that is reliable. In this case, depending on the type of experiment, you could either use distilled water, deionized water, deionized water, ultra pure water. So we have a dedicated room where we produce these types of water so that our results would be very reliable and subject to proper reproduction. This is our genomics lab, like I mentioned, where we have our PCR machines, our sequencers, our iScan machine. This is our qPCR machine, which we used for our COVID-19 testing, and then which is what we use most of the time for our gene expression and a lot of other diagnostic tests. Like for hepatitis B and hepatitis C, viral load testing, HIV viral load testing. This is our sequencer. This is our cell culture room where we do where we culture majorly mammalian cancer cells to do experiments on them. I have mentioned the couple, a couple of the cancer cell types that we currently have. We also, one of the challenges of doing research in Nigeria is problem of constant electricity supply. We've had from 2019, we've had terrible experience in terms of trying to maintain constant electricity in CAMRET, not only for our experiments, but more importantly for the samples that we store and our reagents. Now, if we don't properly store our reagents well under the right temperature, we risk losing the integrity of those samples. And at the end of the day, no matter how good our experiments are, we will not get results that are reliable. So what we have a dedicated 250 kVA generator, but then putting that generator to constant use is very, very expensive. So what we've tried to do is to have a couple of alternative power systems. These are majorly inverters, solar panels, and batteries that we use to power our minus 80 degrees freezer, our other refrigerators and freezers, and also power our experiments. So we've also had our share of problems with theft and other issues of vandalization. So one of the ways that we've tried to overcome that is to ensure that we have proper security. We have dedicated security personnel, and we've also complemented that with CCTV cameras just to see what happens around, and then to try to serve as a deterrent for people that are thinking of engaging in any criminal activity. So in terms of our source of funding, our major source of funding has been research grants. Over the last 5-6 years, we've enjoyed a lot of funding from TED Fund and especially from the National Research Fund. We've also received funding again from TED Fund, but then in this case, it has been majorly other funding, other types of grants that we have received from TED Fund are usually special interventions. We've got two during COVID-19. One was to look at potential therapies for COVID-19. The other bigger one was to actually look at try to develop a recombinant DNA vaccine for COVID-19. We also do engage in short-term research trainings, and that also helps us in generating some funding. This is mostly through workshops. We also do personalized trainings whereby individuals that are interested in learning how to do cell culture experiments or learning how to operate a flow cytometer, or how to extract nucleic acids, how to do PCR. They can also come to CAMRET for personalized or customized training programs, and then we charge for this. We also conduct other diagnostic and research services, which also helps in generating some funding for the center. So this includes diagnostics for hepatitis B, hepatitis C, HIV. We also do provide bench space for individuals that want to carry out their experiments, especially trying to leverage on some of the labs we have, like our Drosophila lab, or the other equipment that we have that they want to use. If they approach CAMRET, we charge a fee, and then we give them access to CAMRET for a minimum of 6 months to carry out their experiments in the center. We have just recently also built a befitting an animal facility. So we also take in researchers, students and researchers to carry out their experiments using rats, mice, or guinea pigs. So in terms of what I consider our strength, we have we have received funding to the tune of over 700 million naira. From this is majorly from TED Fund since 2019, which I would say is one of the best for a center in the university and even across the country. Because by Nigerian standards, this is actually huge. This may not be huge when you consider what happens in other countries. TED Fund does a lot in terms of research funding, but there's still a lot more that needs to be done in terms of funding research in the country. I worked in China in 2016, and in a single year alone, my lab got funding to the tune of $5 million. This was just a single year funding, and I would tell you, my university was a mid-tier university in China, and so you would imagine what those top-tier universities or other universities at the mid-tier level also receive. So, but then TED Fund does a lot in terms of funding locally, but then, like I said, a lot needs to be done. So we have received a lot of funding. And then what we try to do what we've been trying to do to change the narrative in terms of conducting research in Nigeria, using our funding that we get from TED Fund, is not just to use the funding that we get to carry out the experiments or the projects that the funding has been given for, but then we try to actively recruit students to work on all our projects. Now, we understand that postgraduate students are expected in Nigeria to take out money from their pockets to fund their research projects. And I say that is not a sustainable model. That is not a reliable model, because at the end of the day, if you have to rely on a student, a master's student, a PhD student to sponsor his or her research, then they would definitely want to cut corners, do the barest minimum, and just graduate. So if you want MSc and PhD students to do very good research and to evolve into truly independent researchers, then they're funding their research, and funding their research is a must. So that is what we're trying to. That is part of the narrative that we're changing in CAMRET to ensure that every research grant that we get, we maximize its use, not just to carry out the experiments and give the result, or give at the end of the project we submit our report to TED Fund. We also ensure that students benefit, other researchers also benefit from the research grant by employing them to work on it as part of their MSc, as part of their PhD, project. Again, one of our strengths is the fact that we're a leading biomedical research facility in the country. And this is easily, I mean, you can easily see this from some of the things that I mentioned regarding the availability of some of the equipment we have. We have staff that we have research associates that have gone to some of the leading universities and research labs around the world. They've trained and they've come back to contribute their own quota to the development of this of our research center. We've had people go. I mean, we've had people train in the best universities in Asia, the best universities in Europe, and in US. Some of these research associates have gone for their PhDs, MScs. Some of them have just gone for fellowships, and we have a pool of talented researchers in the center. Again, like I said, we have, in addition to our storage facilities, which are arguably some of the best that we have in the country, we also have a functional cell culture tissue lab, which enables us to do a lot of experimentation, a lot of in vitro experimentation, which is not possible in a lot of labs across the country. Yes, there are physical structures meant for cell culture, but most times these cell culture labs are not functional because of one problem or another. We have been able to sustain ours. Again, we have had a very good research. We've had very good research output over the years. Consistently, we only publish in some of the best journals in our fields. Okay, we try to only publish in journals that have impact factors. I know there's a debate for and against the use of impact factor as a yardstick for measuring the quality of journals, but then, I mean, most universities and top universities and research institutes around the world still stick to this metric. And I want to believe it's because there currently isn't a better metric to help measure how reliable a particular journal is. So what we have currently is the use of impact factor. Of course, I understand that other indices are currently being developed, but because we don't have a better one, that is what we use also as our yardstick to try to compete with our colleagues coming from any part of the world. We also, I mean, we have shown prudence in terms of managing the research funds that we have got from TED Fund and from other donors, and that is why we actually have been consistently receiving additional funding, because we're able to prove that whatever funds that we get, we use them judiciously. We also have a lot of collaborations with top research institutes and universities in the country and outside outside the country. I did talk about our MSc and PhD programs that we have. Whatever research grant that we get, we try to use that as an opportunity to help train and mentor MSc and PhD students. Not necessarily because it is one of the indices that is being used to monitor the project, but we just feel it's a duty upon ourselves to ensure that we use whatever opportunity we have to train train the next generation of scientists. So in terms of some of the challenges that we have, I think we still have challenges with visibility. We've tried to ensure that we have a functional website and functional social media sites, but we still think a lot more needs to be done to improve the visibility of our center within and outside the country. And we're actually working very hard to see that this happens. That is why we're keeping our sites very active. We try to update them. We try to update them regularly. Of course, we do have operational hiccups here and there, which are some of the things that we try to work on. For example, one of the things that we have tried to do is to make sure that there's sustainability to some of the things that we do. And like I mentioned, that is why we have created dedicated website, phone number, and email for the center. But then in terms of how we do our procedures, we think that there's a lot more that can improve. Now, one of the challenges that I see, or one of the problems that I see, especially within the university system, is whenever, and I think this is something that needs to change, not just for CAMRET, but for most research or most university units across the country. Once an individual gets an appointment, gets appointed to a particular position, there barely is any particular guidance or any particular template for how that particular role needs to be executed. So most times the individual is left to trial and error to sort of navigate the way through that particular position. And I think this needs to be changed for there to have sustainable impact for there to be sustainable development in our university system. This needs to change. In terms of funding, we have done arguably very well, but then we still need a lot more funding. Like I said, what TED Fund is doing is quite commendable, but if you compare this to what obtains in other parts of the world, this is very, very meager. I was looking at the literature, I was looking at the data for research funding, and I did a comparison between as of 2019, the data for 2019. As a 2019, TED Fund was celebrating the fact that it had given a funding of 1 trillion naira over 10 years. That is from 2009 to 2019. TED Fund disbursed a total of 1 trillion naira for research grants across the country. And then I did a calculation to convert that to dollars. As of 2019, $1 trillion was 1 trillion naira was just $1 billion. Now, the US alone, or China, each gave its researchers in the country a total of more than $500 billion just for research in 2019 alone. In 2019 alone, China or USA gave out a total of each one gave out more than $500 billion to its researchers, while TED Fund was celebrating giving out $1 billion over a span of 10 years. So like I said, TED Fund is doing wonderfully well, at least within the Nigerian context, but then a lot more needs to be done. So in terms of CAMRET, for example, we look forward to getting additional funding to equip our animal facility. We have just built an animal facility, but we need equipment in that place. We need more training to be done on some of the skills that we lack, for example, in terms of animal handling, animal experimentation. We have the current space that we have for CAMRET is quite small, so we are still looking for we're looking for an opportunity to expand, which will be very soon, hopefully. We're also looking to purchase additional new specialized equipment, because as we grow and as our projects expand, we notice that there's the need. We're doing well currently, we're doing a lot, but then we notice that there's a lot more expertise that we need to bring into our experiments. We need to include other types of experiments in our projects, and we can only do that if we have the befitting, if we have the equipment to do that. Of course, we have issues sometimes with alternative power supply, especially for like our minus 150 degrees freezer needs, just like we have done for our minus 80 degrees freezer, it needs to have constant power supply through alternative means, using solar panels, inverters, and the likes. Of course, the issue of sustainability is always on our minds. What happens in the next 5 years? What happens in the next 10 years? And we are, although this is a challenge currently, but we're still looking for a way to make sure that CAMRET stands on its feet, so that we don't have to rely on external sources to be able to operate. By the way, even currently, CAMRET doesn't receive a dime from any from the federal government or from the university. We are semi-independent in the sense that although we're part of the university, but we don't receive any funding, not even in the form of DTLC that is given to other units of the university. We don't receive that. We don't receive any sort of funding. So we do everything that happens in CAMRET, or everything that we do is based on the funding that we get, and based on what we get in terms of research funds, and based on what we get from the services that we offer. So these are some of our collaborations that we've worked on our projects in the past, and still working with some of them. We have active collaboration with Nigerian Institute for Medical Research in Lagos, and the NVRI VOM in Plateau State, NARIT in Zaria, Kaduna State. We also have active collaborations with University of Jos. We also work very closely with Prof. Peter from University Hospital in Frankfurt, Germany. Prof. Samuel Temesgen is also one of our biggest collaborators from the USA. In terms of our collaboration with Prof. Peter in Germany, we were recently in the news for what we were trying to do with his team in Germany. So this is the project that I mentioned earlier regarding testing for cases of relapsing fever from patients that have recurrent fever in Nigeria, and we were able to see that a lot of patients have have the pathogen that causes a relapsing fever, which is Borrelia species. And we're still working on this project. We're looking to expand in future. Some of our potentials, looking into the future, what we see as our potentials are, we actually, because we now have been able to domesticate the technology and the processes for the development of a recombinant DNA vaccine. We have done that for Lassa fever vaccine. We've done that for COVID-19, the COVID-19 vaccine. We were able to develop a candidate vaccine that is currently undergoing preclinical testing for both COVID-19 and Lassa fever. So we see ourselves as major players in terms of development of next generation vaccines within the country. The importance of this is that if you reflect back on what happened during the COVID-19 pandemic, when there were a lot of disruptions in supply chains across the world, even if you had the money to purchase drugs or diagnostic kits or vaccines, it became very difficult to actually purchase those diagnostic kits or therapeutics to your country. So if we domesticate this in Nigeria, and we have a whole system that is able to develop to test or to develop the recombinant DNA vaccine candidate, to test it, and then to mass produce it, then that would be a very big win for the country. And we see ourselves as major players in this particular process. We also see ourselves as players in development of diagnostic kits, which we have experimented on, and we're still experimenting on. We also have the capacity to be involved in screening for veterinary diseases, especially because we sit at the border between Nigeria and other countries. Our neighbors to the north, where there's a lot of movement of animals and humans, and so what this means is that a lot of diseases can be spread across the border. And our position as a reference lab for the Nigeria Center for Disease Control, we actually see ourselves as major players in screening for diseases during pandemics or epidemics. Now, because we have a minus 150 degrees freezer, a cryogenic freezer, which can be used for biobanking, we actually see ourselves as a major player in biobanking to store precious samples, not only from within Sokoto, but from across the country. We are able to store precious samples in the form of sperms, in the form of eggs, or any other type of tissue that an individual wants to store for an extended period of time. We're also considering going into the manufacture of basic laboratory reagents, again the problem is, in this case, the issue is, or the challenge is similar to what I mentioned regarding what happened during COVID-19 pandemic, whereby it's difficult to bring in some consumables or some therapeutics or diagnostic kits. In this case, it still is a problem to bring in some research consumables or reagents into the country, and we are in the process of trying to manufacture some of the basic buffers and some of the basic reagents that we can produce in the country to reduce the importation of some of these consumables into the country. Of course, one of our biggest businesses is the training of next generation of scientists. Just like I said, we leverage on any research grant that we have to try to trained MSc, PhD, and any other researcher that is that has the motivation and that is willing to stick to the rules at CAMRET. Thank you very much, and I'll open the floor for Q&A.
Thank you very much, Professor Imam, for that wonderful presentation of CAMRET. And I think the way you started, not only CAMRET, you have said a lot about our university in general. I think we are going to launch into the question and answer session. If anybody wants to ask a question or want to make contribution, you can either raise up his hand so that he can be given a chance to talk, or he can write his questions on the chat button. I can see that is one.
It will feel okay as a father mobile because.
Okay, so there's a comment here. There's a comment that says it's not really a question, but a comment saying, I'm suggesting that in order to overcome some of the challenges faced by the center, the university should provide an office that comprises dedicated, or I guess he means that is dedicated to managing database and social media issues such as ICT agents. Well, although this is a this is a very good idea, but the whole idea is, like I mentioned earlier, CAMRET is semi-independent. Semi-independent in the sense that it's established to be able to stand on its feet. So a lot of the staff we currently have over the research staff that we have are about support staff, probably about maybe 8. But then only 2 of those are employed by the university. All the rest are employed directly by the center. And I think this is one of the ways to go. Why? Because we have seen that a lot of our research centers and a lot of our agencies in the country are unable to operate well because most times they depend on government for handouts. And then the reality is, if you depend on government for handouts without trying to be productive or trying to stand on your feet, most times the end result is that you end up not putting in the effort that you can actually put to make the place sustainable. But because you realize that even if you don't put in any effort, there's there's money or funding that will come from government. Then most institutes or most agencies tend to be lazy. So CAMRET is the whole idea of CAMRET is trying to change the narrative in terms of how research centers operate in the country. And so far, we're actually, yes, we have a few challenges because of funding, but we're very happy with the position we have, the semi-independence that we have, and then how we're able to do things without active involvement or without active funding from the university, because then we're able to chart our own course to actually determine in what areas we want to pursue, what areas we want to invest in if we have our own funding. So yeah, we may consider already we have part-time staff that are involved in managing our database and social media sites. That is not done by researchers from within the center. We have people that are part-time staff employed, paid by CAMRET to do this. But then having dedicated staff to do this, I don't think that is what the university wants to do. Besides, there are not enough staff in the university to even go around to support our major activity, which is research. So I don't think the university will be able to provide staff to just to maintain database or to handle social media accounts. Thank you.
Thank you very much. I think Dr. Aminu Shitu, I can see you raising up hand. Please go ahead, unmute yourself and speak up.
Yes,
Thank
please.
you.
Thank you. Yeah, thank you so much, Dr. Mani. Am I audible, please? Hello.
Yes, you are. Yes, go ahead.
Oh, yeah. Okay. Thanks to Professor Mustapha Umar Imam for a wonderful presentation as usual and for telling us everything about CAMRET. Initially, I actually thought most of the research at CAMRET are more to do with non-infectious diseases, chronic diseases, and nutrition. Well, I was a little bit skewed to Professor Imam's area of interest. So I was thinking like most of the research skewed towards that and also looking at more of publications on that area. But I'm happy to hear that there are also a component of infectious diseases, which is quite interesting, and that makes the CAMRET a very good and interesting research institution. And with looking at the mandate of CAMRET, which is like 2-in-1, advanced medical research and also training, I would like to ask a question, or I would like Professor Imam to comment on the training aspect. What I mean by training aspect is that assuming someone who believes or who thinks that he has an expertise on a particular area which he is willing to share, and then there is a possibility of discussing it with the management, and then if that could be granted, and then somebody can come in to share some ideas with the research students at CAMRET. Is that possibility or chance to do that? Thank you very much.
Okay, thank you very much, Dr. Aminu, for that question. And I'll try to answer your question together with the one that has just been asked by Professor A.A. Magaji in the comment section, which goes like, I had more research than training, which is part of the mandate of CAMRET. How often do you engage in training? What is the cost like? And what are the areas, please? Okay, so generally in terms of training, we do a lot in terms of training. If you recall back on, if you reflect back on what I said regarding using every research grant that we get to try to support the studentship of MSc and PhD candidates, that in itself is training. We're doing that because we've taken into consideration that one of the core mandates of CAMRET to ensure that we don't only do research, but we also train students in the process. So we have been able to, using our research grants, we've been able to train, to graduate close to about maybe 10 MSc students and about 3 PhD students so far. We still have a lot more. We still have a lot more currently in CAMRET. By the way, CAMRET in the past did not have the capacity or was not able to take in students directly from through the PG school, that's the postgraduate school. So what we did in the past was to work together with departments of the university to the departments will admit the students, and then the students will get sent to our center for their research projects. Of course, they're going to be domiciled in our center for the entire period of their studentship. Most of this we do with departments that we have direct linkages or collaborations with. For example, myself, I am from the Department of Biochemistry, so we do take in a lot of students from biochemistry. And part of the reasons why we work with departments that we have linkages or direct collaborations with is for practical purposes. Practical purposes in the sense that we want to make sure that we have, we take in students from departments that we can easily have a say on how, how those students, how the project of those students is handled, or how their progress is actually being assessed. So yes, that is one of the ways that we train students, MSc, PhD candidates. So again, in terms of the customized training programs that we have, so we try to do trainings every year. We have training on cell culture, and then we have training on the use of flow cytometer, and then we have training on basic molecular biology skills. So in the past, what we did was we had a training program whereby we combined cell culture experiment, a workshop on cell culture and flow cytometry, and we charged 150,000 naira for a 1 week training. And this was a hands-on training. So what it meant was every single participant that came for that training actually had the opportunity to practice some of the skills that were being taught. So in the future, we're also looking to expand to other types of research trainings. But currently we only conduct 2 types of research trainings, customized research trainings. That is on cell culture experimentation and flow cytometry and basic molecular biology skills. Although we organize these at regular intervals, these are also open to individuals to actually approach CAMRET at any time, and they will be given this training for an entire week if they pay the required, what we're asking for in terms of payment. So I hope I have done justice to this question on training.
Yes, you have, Prof. Thank you for the good attempt.
Okay, sir. And then maybe one more thing regarding what Dr. Aminu Shit was asking. If yes, we're open to getting, if there are, of course, there are areas that we don't have the expertise on. So we're open to receiving collaborators, anybody with the skills to be able to train our students or to train other researchers on any field. We're open to discussing that idea and then giving them the platform to train our students or to even train other researchers from outside our center or outside the university, outside our department. Thank you very much.
Thank you.
So do we have any other comment or question? Okay, so Dr. Mani. It appears the host is having an issue with his network or connection. Any other comments or questions, please? In the absence of any, perhaps we can call it a day.
Okay. Thank you, Professor Mustapha, for that wonderful presentation. And I think we don't have more questions from participants. I think before we call it a day, sir, I would like to invite either the Director of Research, Innovation and Development, or any of his assistants to give us a closing remark. Professor Bayawa, are you there, sir? What of Dr. Malami? Dr. Malami, can you hear me? Okay, I think Dr. Malami is also unavailable, and I think we will call it a day as Professor Mustapha rightly said it.
Dr. Mani,
we thank everyone for
Dr. Mani, sorry to interrupt. Sorry to interrupt.
Okay, sir.
Yeah, so somebody is asking if they can have our email. So I have just put our email in the comment section. We have 2 dedicated emails, one for the center and one that was given to the center by the university, so we can be reached via any of these emails. Thank you.
Okay, thank you, Professor Mustapha. I want to thank everyone for attending this important webinar, and I think, as we have decided at the beginning of the webinar, that due to the low attendance, especially at the beginning, we are going to distribute the recorded webinar far and wide, because for me, this is a very important center that has the potential for improving research not only in Nigeria but the world over. So I think users or those that attended are likely going to be sent the recorded webinar, and especially for those that were unable to attend. Thank you very much. And I think at this juncture, we call it a day. Bye-bye.